Hello, myself Lokesh Sharma. I am from room 317. So, my first topic is molecular biology and physics. So, what is molecular biology and physics? So, now we talk about the definition. It is the study of biological molecules and their in living organism, or the study of matter, energy, and fundamental forces of nature. What are the key concepts? Key concepts are DNA, RNA, proteins, enzymes, cellular processes, or force, energy, mass, motions in thermodynamics is also a part of the molecular biology and physics. What are the techniques we used in biology and physics? Techniques are PCR, gel electrophoresis, sequencing, microscopy. Fundamental units are nucleotides, amino acids, and cells. Now, what is DNA? RNA is also included in the molecular biology and physics. Now, what we talk about the DNA and RNA, it is the genetic information storage and transfer. Proteins are also included in it is a performs most functions in the cells, that the proteins are help in the function of the cell. Now, the gel, what is gel electrophoresis? It is the separate nucleic acid or protein from the DNA. Fundamental units are nucleotides. Nucleotides are the building blocks of nucleic acids like DNA and RNA. Amino acids. Amino acids are building blocks of proteins. What are the major discoveries in the molecular biology and physics? They are the structure of DNA by Watson Crick in 1953 and discovery of genetic code and its role in the protein synthesis. Newton's law of motion in 17th century and Einstein's theory of relativity are the major discoveries in molecular biology and the physics. Now the current trends. What are the current trends going on? It is the CRISPR. It is a technology for gene editing and synthetic biology for designing new biological parts, personalized medicines based on the genetic profiles. Quantum computing, revolutionizing the information processing and renewable energy technologies to combat climate changes. Now, what are the changes facing molecular biology and physics? The challenges are complexity of biological systems makes predictions difficult. The data management in genomic due to the large data sets, understanding dark matter and the dark energy role in the universe, and the last one is addressing climate change through sustainable practices. What are the future direction? Future direction are advancement in the gene therapy for the treating genetic disorders and development of the novel vaccines using mRNA technology. Now my second topic is nucleic acids. What is nucleic acid? Nucleic acids are polymer of nucleotides, and nucleotides consist of nitrogenous base plus pentose sugar plus phosphate. Nitrogenous sugar. Nitrogenous sugars are basically divided into two, purines and pyrimidines, where purines are adenine and guanine, where pyrimidines are cytosine, uracil, and thymine. Thymine is present in the DNA and uracil is present in the RNA. If we talk about the sugars, sugars are pentose sugar. Pentose sugar is present in the RNA, and here is the structure of the ribose sugar. And deoxyribose, it is present in the DNA, and here is the structure of the DNA. Here, when this ribose sugar is attached to the nitrogen base, this H is removed here. Now the phosphate. Phosphate is also called as a phosphoric acid. Here is the structure of phosphate. When this phosphate is attached to the ribose sugar and nitrogenous base, then there is this H is removed under certain conditions. Here is the structure of deoxyguanine monophosphate, where this is the guanine structure, this is the pentose sugar, and this is the phosphate group, where the base plus sugar is made nucleoside, and base plus sugar plus phosphate, they combine form nucleotide. Here we already studied about the AMP, ADP, and ATP. The AMP cause adenosine monophosphate, ADP is adenosine diphosphate, and ATP is adenosine triphosphate. Now, if we take the base adenine, here nucleoside formed is adenosine and nucleotide is adenine cyclide. Same as if we take the guanine as a base, the nucleoside formed is guanosine and nucleotide is guanylic acid. Same as cytosine, the nucleoside is cytidine and nucleotide is cytidylic acid. So now, if we talk about the ribosome, as we know, the ribosome is an enzyme which is also a nucleic acid is act as a catalyst. Now here the structure of the DNA where the guanine is attached with the pentose sugar and the pentose sugar is attached to the phosphate. So here the whole structure of the DNA. Now as we know, the DNA have two strands, 5 prime to 3 prime and 3 prime to 5 prime. In DNA, A is attached to the adenosine is attached to the thymine with double bond, and guanine is attached to the cytosine with